Good morning, and thank you very much for the organizers to give me the opportunity to present this work. And in the first half, I'm really going to talk about the genetics of SCN2A, and then in the second half of the talk, I would like to move beyond SCN2A and really think about concepts and differences, how we think about SCN, about the channelopathies, SCN1A, 2A, and 8A, and how we make sense of the different phenotypes. And that's really following on the questions from the last session and quite fitting into this symposium. So to start off with SCN2A, I've got no disclosures. So SCN2A is the sodium channel alpha 2 subunit, and it's a watch-gated sodium channel and widespread expressed in the CNS. And its main role is in the initiation and conduction of action potentials, and mainly expressed in axonal initial segments and nodes of Ranvier. And most importantly, it's critical for normal early neuronal development. And it's a transmembrane protein, and it has got 4 domains that fold up together to build a pore. And what's important here is that we've got certain areas that are particularly important, which is the voltage pore, voltage sensor here in yellow, and then the pore region in pink. And we've learned quite a lot about SCN2A-related developmental disorders from our previous speaker, and this is just to illustrate the different ways this does present. It presents clearly with a phenotype of epilepsy, and there's been some gain of function effects, and this is in a mouse model in mouse by Yamagata, recently showing an expression of SCN2A in the hippocampus as well as in the neocortex, but it's also associated with features of autism spectrum disorder. And recent work by Ben Shalom showing that this is mainly associated with loss of function variance. And finally, the spectrum is widening, and we also now have features of a movement disorder and episodic ataxia. And Dr. Wolf already alluded to this, and Holger Lerche is going to talk about this a bit more, but clearly we all could see that in the mouse there's clearly expression in the cerebellum as well, and gain of function effects in the variance underlying these symptoms. Now, Marcus has really eloquently alluded to the 4 different groups, how the children with SCN2A variants present. And so now we're going to look at the genetic basis of how we explain these different variants. So the variant classes we find in SCN2A are mainly missense variants. So to 83%, these are missense variants. This is where one amino acid is exchanged for another amino acid. And what we know is that from the previous presentation also that the phenotype associated with this is really varies a lot. So from very benign phenotypes to severe phenotypes. So it's really a question about if we have a missense variant, how might this present later on? And we know that overall about 17% are associated with truncating variants, and these are nonsense frameshift, splice site, and insertion deletions. And previously, one might have thought if you have a truncating variance, this results in a very severe phenotype. However, the story here is a bit more complex, and we're going to explore that a little bit later. I've now really divided the variant subclasses by the phenotype, and what we can see in the benign familial neonatal infantile seizures and the early onset epilepsies. So these are exclusively missense variants. If we then look at the late onset epilepsies, there's still majority missense. However, the fraction of truncating variants is more frequent, about a quarter. And if we then look at those presenting with intellectual disability and/or autism spectrum disorder, then the proportion of missense variants is only a quarter, and 3/4 are actually truncating variants. Now, looking at the types of SCN2A variants in patients, so the missense variants are mainly found to have present with seizures in 78%, and these have seizure onset in the first weeks of life. And only 20% have a clinical diagnosis of autism. Autism symptoms are being reported. And looking at the truncating variants, about 50% of those will also have seizures. However, these are seizures after year 2 of life, and the majority have a clinical diagnosis of autism or autistic symptoms. And what's really important to bear in mind is that not all missense variants actually cause disease. So if we look at normal population, you will find a certain fraction of SCN2A being present as a missense variant and not causing disease. So SCN2A channel is made out of 2005 amino acids, and we know of over 400 amino acid positions that are mutated in 140,000 adults without severe forms of epilepsy, and we know of over 100 amino acid positions that have been reported in neurodevelopmental disorder patients. And this, from my collaborator Dennis Lau, has been in this group. They've been really looking at trying to explain this graphically. So this is a representation of the SCN2A missense variants found in the general population. So at the bottom graph here, you've got the representation of the SCN2A protein. So again, with the voltage sensor in yellow and the pore in red. And this is from amino acid 1 to about 2,000. And you can see there's the 4 transmembrane domains, but you can also see there's these big intracellular loops where we think that their function is really not that important. And here you've got mapped against the protein sequence, you've got the missense burden. So this is from a database of the general population and the SCN2A variants you find and the frequency of them. And what you can see is there's clearly peaks of missense variants present in the general population, which correspond with this area of not so strong importance. So clearly you've got missense variants also that are not disease-causing. So how do we differentiate variants? So for truncating variants, this appears to be straightforward, that we have a loss of protein function, and therefore this will present with some effect. However, if you have missense variants, this is often much more difficult to classify, and it will depend on whether the variant might be Denovo or whether it's inherited. Does it affect a functionally important part of the protein? Is it conserved and related to species? Are there differences in amino acid composition? And are there any functional data available? And I would now like to show some a slide how we look at, for example, differences in 2 amino acids. If one amino acid is exchanged for another one, and there clearly are some physicochemical differences between amino acids. So the amino acid structure is similar to all amino acids. So you've got amino group and carboxyl group. However, you've got a side chain, and the side chain really defines that amino acids. And you can try to quantify and characterize that side chain characteristics by looking at polarity, molecular volume, and composition, and putting all this together gives you a score which is called the Grantham score, and that quantifies the difference of amino acid substitutions. And if you've got a score of 0, there's no difference. However, if it's got a major difference, it's up to 215. So some time ago, we looked at the genotype-phenotype associations across the voltage-gated sodium channel family, so really looking at effects that we see across SCN1, 2, 3, 4, 5, 8A, and so forth. And here really focusing on SCN2A, and what we found was that for SCN2A, when we looked at the different phenotypes, so this is the benign familial neonatal seizures. At that point, this was in 2014. There were 11 cases, and of the more severe phenotypes of Ohtahara syndrome and early epileptic encephalopathy. So what we found is that the Grantham score actually was clearly different. So the amino acid change in the benign phenotype was much lower, significantly lower, compared to the more severe phenotypes. And I think Holger's later presenting some functional data also to show some differences. So this is clearly an interesting observation across this cohort. Now, how we eventually make sense of all the genetic data and interpreting those. We do this in Glasgow by having an epilepsy genetic multidisciplinary team meeting. So this means that child neurologists such as myself and Professor Samir Zuberi, as well as our geneticist Sheila and Daniela, and our lab scientists, we sit together every week to discuss the results of our gene panel. We've got 104 gene panel, so all the new results we have available every week, we discuss their significance, because clearly there's lots of factors to take into account. And I think the role of a physician really is to give the perspective of the phenotype to the scientists, and this is a great learning exercise as well, because our lab scientists are really interested in hearing more about the phenotypes and vice versa. So I will now talk a little bit more about the specific phenotypes found in SCN2A and the genetic changes. So for the benign familial neonatal infantile seizures, 82% of those tend to be inherited. There were only 6 Denovo variants found, and these tend all to be missense gain-of-function variants with generally a lower Grantham score. And as we already heard earlier, there's different subgroups emerging, and particularly one with later evolving episodic ataxia due to a gain-of-function variant, and Holger's group showed this very nicely, and there seems to be a particular hotspot, which is here, but I'll leave Holger to explain that later in his talk. Now, if you look at the early onset epileptic encephalopathies, these tend to be all de novo variants, and they tend to be gain-of-function variants in patients with early infantile onset epilepsy. And for an example, as a gain-of-function variant, so if the channel cannot close, you might get too much firing of the neuron, resulting in a gain of function. And if you then look at the late onset epileptic encephalopathies and autism spectrum disorder, those tend to be variants associated with loss of function, and particularly an example would be if for example, the channel cannot open, you might not have, or the channel is not expressed, you might not have enough firing of the neuron, resulting in a loss of function. And this is work by Ben Shalom. So when they looked at individuals with autism spectrum disorders with SCN2A variants, they all tended to be loss of function variants. And I would like now to move on, really looking also beyond SCN2A, and with my collaborators, Dennis Lau, and also Ricky Möller, who's in the audience as well, looking at systems and how can we explain the heterogeneity that between the different phenotypes, SCN1A, 2A, and 8A, and most recently also SCN3A. So what explains the phenotypic heterogeneity among sodium channel neurodevelopmental disorders? So what's really important to notice is that these all evolved from common ancestry. So there's a clear evolution of voltage-gated iron and sodium channels, and we have here the monomers of the potassium channels, and then moving on to tetramers in calcium and sodium channels. And we know that those expressed in CNS are very closely related. And Katrol's group has shown this very nicely. And this is just a representation of the sodium channel subunit gene tolerance for missense and loss of variance. So it's a figure to show you how well tolerated a specific variant is actually in the general population. So these are data from over 60,000 individuals from the exact database. And what you have here on the x-axis is the Z-scores to suggest that genes are intolerant for any missense variants. This is down here, so these, if you're intolerant for missense variants, you verge towards here, and on the PLI scores, if you're intolerant for any truncating variants, you end up here. So what's really important is this group over here, in the top right corner, is intolerant for any missense or loss of function variants. And that's really important because these are all the genes that we're interested in. So SCN1A, 2A, 8A, 3A, and SCN5A as well. So these are genes, if you find them in an individual, the likelihood that this is disease-causing is quite high. If you compare it to all the other variants, for example, a good example is SCN2A. SCN2A has got a clear role as in pain disorders. However, the role in epilepsy is very questionable. And from our own practice, we come across so many SCN2A variants where we eventually have to say, well, actually, we don't think this is disease-causing. So it's an interesting way to look at many variants we find where you have to have, there's a caveat, yes, they're known in particular diseases to be disease-causing. However, you will also come across lots of variants where you think, you're not quite sure, is this disease-causing or not. However, with SCN1, 2, 3, 8A, all of those, the likelihood for them to be disease-causing is much higher. And just moving on to the SCN1A, sodium channel neurodevelopmental disorders. So we think that 1 to 2% of patients with neurodevelopmental disorders carry a Denovo variant in either SCN1A, 2A, 3A, and 8A. And that is a significant proportion. And we know that the phenotypes are heterogeneous within genes, and it's not always obvious which patient fits the gene, and there's lots of overlap as well. And so the interpretation of genetic test results in these genes is still in its infancy, and very often it's very challenging. So these are data from ClinVar, which is an open source, where it's characterized whether a specific variant is pathogenic or not pathogenic. And if you again look at SCN2A, so you have, yes, there's a number of variants where we know they're benign or we know they're pathogenic. However, in the middle, you still got a significant number of variants of unknown significance. And we really need better tools to characterize this better. So what we did as a collaboration, we had an aggregation of patient clinical and genetic data from large international cohorts and the literature. And really, to take this work forward, you need collaboration among different groups. And so we contributed the SCN1A variants. Vicky Muller's group and Markus Wolf's group presented the SCN2A variants and the SCN8A variants. And we also looked at copy number variants. And we also collected and generated RNA data sets, and this was partly human whole brain tissue gene expression mapping from the Allen Brain Atlas, and we also looked at human and mouse single-cell brain tissue, and this was provided by our collaborating partners, and we developed bioinformatic methods to compare missense variant distribution. So I think what's fascinating is to look at the developmental expression of sodium channels, different sodium channels. So, and there clearly is a difference here. And if you, for example, here look, so we've got in the first box, this is prenatal, then infant to childhood, older than 14 years and older than 21 years. And if you focus, for example, on SCN2A in orange, so we know that even prenatally, this is expressed early on. And this is fascinating. There's a recent case of an SCN2A variant, a child presenting in utero. So this is very early presentation and expression, and it remains expressed throughout. If we, for example, then look at SCN1A, it's not expressed, early on. However, its expression then rises. If you look at SCN8A, that similar expression rises. However, with SCN3A, interestingly, it's expressed very much prenatally. However, then the expression tails down. And another way of looking at whether certain genes and channels are co-expressed. So here we looked at the gene correlation of cell-type specific expression of sodium channel subunits in the developing human midbrain. And this is looking at published RNA sequence expression data. And what you can see here is that wherever you've got a very strong correlation of co-expression is in dark blue. So whatever is dark blue is strongly correlated. And here you have, for example, you've got SCN1A, obviously co-expressed with SCN1A. Then you've got SCN3A, 8A, 2A, and 3B. And what you can see here clearly is that SCN1A is not correlated with SCN3, 8, or 2A. However, if you look at SCN3, 8, and 2A, they're all co-expressed together, which would fit with the hypothesis of being gain-of-function variants, and here excited being expressed in excitatory cells, this being expressed in inhibitory cells. So clearly there seems to be an expression of SCN2A, 8A, and 3A, and correlation for expression in developing midbrain. And this might also be important if you think about if one of those channels might not be working properly, whether there might be compensation by other channels that, let's go back to that. So you might think if, for example, SCN2 or 3A is not expressed properly, however, that might be able to be rescued by a co-expressed channel, and this is clearly something that we can discuss later. Now, SCN2A is expressed first, followed by SCN8A, and lastly SCN1A in human brain development. This is, again, the data from the Allen Brain Atlas. You can see here, again, embryonic expression up to adult life for SCN2A first, followed by SCN8A, and then by SCN1A later. And we now compared this to the actual age of seizure onset by gene and variant type. And what's really interesting here is, this is a logarithmic scale. So here you've got the different groups, SCN1, 2, and 8A, and then you've got the day of seizure onset. So this is day 1, this is day 10, this is day 100, day 1,000. So this is 1 day, 10 days, 1 month, 1 year, 3 years. Yeah? And what you can see is that those that are expressed earliest also present earliest. So we find the earliest expression and presentation of children with early onset seizures is in SCN2A, which is then followed by SCN8A, and then lastly by SCN1A, and this is for missense variance. Yeah? And if we now compare this, and this is really also the work by Marcus's group, looking at clearly there's, in this group here, we've got mainly gain of function variance, and here we've got mainly loss of function variance, and those are present later. So if we now compare this with the protein truncating variance in those genes, what we find is, interestingly, if you have a truncating variant in SCN1A, it really doesn't change the age of presentation. This is what we all know, that there's no real difference between missense or truncating variants in SCN1A. So they both present at the same time, so it doesn't really matter whether it's a missense or truncating variants. What's interesting is for SCN2A, if you look at the protein truncating variants, they tend to present really late. Yeah? And that's similar to the loss of function variants. And something similar we see here for SCN8A as well. And then we looked at the copy number variance. So this is really looking at if you've got a deletion or a duplication. So if you have a deletion in SCN1A, that again presents similar to missense or protein truncating variants. So the phenotype appears to be similar. However, if you look at SCN2A, what you really find is if you have a deletion, they tend to present later. However, if you've got a duplication, you've got too much gene product, then they present earlier, which is really interesting. So the duplications we see presenting quite early on, similar to the gain of function variants. Now, why does loss of function and of SCN1A and gain of function of SCN2 and 8A cause epilepsy? So, and Katras and Massimo's group very clearly showed that in previous rodent models that SCN1A is specifically expressed in inhibitory neurons, whereas SCN2A and 8A are expressed in excitatory neurons. But is this really the whole story? So these are human data from neuron-specific expression showing that all 4 sodium channel genes are expressed in inhibitory as well as excitatory neurons. So these are single-cell transcriptome analyses from our collaborators from the Macaro lab, showing, this allows the measurement of gene expression via mRNA in hundreds of thousands of individual cells simultaneously, and these are data from over 80,000 single cells from the adult human cortex. So these are human data. And what you can see here is to the left, so you can see a cluster of adult human cortex cells, and they are marked by a neuronal marker. So I'll take you through this. So RBFOX3 is a neuronal marker. So here you've got your cell types, and these 7 cell types are neuronal cell types. And whenever you've got a neuronal cell type, a neuron, and that's colored in blue, you can see that. And then you can look at excitatory and inhibitory markers. So all those cells that are mainly excitatory are here represented in red, and those cells that are mainly inhibitory are represented by light blue. So if you, for example, then look at the expression of mRNA expression of SCN2A, so what you find is that on balance, so you find all cell types expressed. However, you've got relatively more expression of excitatory cell lines compared to inhibitory ones. And this is very similar to SCN8A and SCN3A. So you've got similar expression throughout these cell lines of SCN8A, however, a slightly more expression of excitatory cells compared to inhibitory cells. And this is slightly different for SCN1A. So SCN1A also expressed throughout. However, you've got, for example, particularly for these layers, you've got more expression in inhibitory neurons compared to excitatory neurons. So we're talking about more a balance. So you have expression in different tissues, however, they're not expressed at the same level. And so if we now think about how we put this into a model of, for example, early onset seizures, so we know that all genetic variants in SCN1A lead to seizures due to loss of channel function. There's less inhibition and loss of channel function. However, if you look at missense variants and duplications in SCN2, 3, and 8A, this leads to seizures due to too much channel or excessive functioning of the channel. And we could show that the seizure onset is clearly determined by the gene expression. Those genes, SCN2A expressed first, presents first, followed by SCN8A and then SCN1A. And if we now think about a model for late onset seizures, here we have truncating variants and deletions in SCN2, 3, and 8A, and they might be able to be compensated due to correlated expression of SCN2, 3, and 8A. However, the compensation might be incomplete, and children develop neurodevelopmental disorders with later onset seizures. And another thought on this would be if you think about the I think it's quite striking to see the developmental expression and how this correlates with the phenotypes. And if you think about SCN3A expression, we've now had the first case series of SCN3A, which is expressed early on and prenatally and then tails off. The question is, what role does this play in later onset seizures, whether there might be some rescue mechanisms, and it would be really worthwhile looking into this in the future. So I would now like to look briefly at how can we predict gain versus loss of function regions to facilitate missense variant interpretation. And here, again, this is Dennis and Eduardo who built this tool. So I showed this picture earlier as an example for SCN2A. What is the burden of variance in the general population? If you now compare this to the actual patients, you can see that there's a clearly different spread of variance found. So these variants are clearly clustered in areas of greater importance, and I think we can learn a lot by, and then Dennis is just doing this at the moment, building a tool where you can compare SCN1, 2, and 3 and 8A, as well as other sodium channels, and because there's lots of similarities in expression and where we find disease variants. So, of course, this is only a 2D model. So what would be nice, and Dennis has done this particularly with Sumaya, to build a 3D model around this, which illustrates, so this is looking similarly, where do we find certain variants, which other variants might be closely associated with that. So this is a proximity-based, three-dimensional scanning of the structure model and enrichment of patient variants around each residue within close spatial proximity. And so it would be very interesting to look at, for example, certain patients or certain variants that are reported to respond well to certain medication, and to see, do they actually in the 3D model, do they cluster together? Can we gain any information on phenotypes and which treatment strategies might be useful in this? And you can then build this into a 3D model, where you, for example, might look for certain hotspots for gain-of-function variants that might be associated with seizures in the first 3 days of life, or you might look at hotspots of loss-of-function variants and looking at those presenting with autism features. So this is really work in progress, but I think it would be worthwhile to pursue this, as it might have clinical implication. And I would like to, it's moving. And so I would like to really acknowledge all the different collaborators without whom all this work wouldn't have been possible. So particularly Dennis Lau, who's been instrumental in starting this work, as well as Rike Möller and our collaboration, then the group by Juliana and Eduardo for making all the models, and Steve McCarroll for the transcriptome data, and of course my team in Glasgow. Thank you.